basically a branch of a motor neuron. So here's the branch, the axon collateral, the axon of a motor neuron coming to stimulate this muscle fiber. Each axon ends in smaller branches called telodendria. They interface with the muscle cell. The area where the neuron meets the cell is known as the neuromuscular junction. All this is neuromuscular junction. That's the zone. Here you see a cutaway view of the neuromuscular junction. These are the ends of the telodendria, the little branchlets coming off. These swollen ends are called the synaptic end bulbs or terminal end bulbs. They contain the neurotransmitter that's required to stimulate the membrane of the muscle cell. That's what we're talking about in lecture. This white edge, that's the edge of the sarcolemma, the membrane, and it's folded in and in. Those are the motor end plates that the neuron fits into. Bless you.
Thank you.
Right? The motor neuron doesn't actually physically touch. You see the little pink space in between. That's the synaptic cleft, the space, excuse me. This is inside the cell. These gray things are mitochondria. This is inside of the muscle cell, so this is generally sarcoplasm, the inside of the cell.
That white stuff? I mean the yellow stuff?
Yeah. Okay. Wedged here in between the endomysium and the sarcolemma, this is the myosatellite cell. This is a tiny little stem cell that lives on the outside of the mature muscle fiber for repair and regeneration. Once we go inside the membrane, these are the organelles. Mitochondria, there'll be Golgi apparatus, there'll be ribosomes. I mean, all the organelles of a cell are in here. They don't have them all here, but they're in here. They show you mitochondria. They show you one of many nuclei. What they're emphasizing, though, are the different organelles not found in other cells, the organelles for contraction. And that's these guys. This is a myofibril, a myofibril, a myofibril. These are the structures made up of actin and myosin, meaning thin and thick. And now you can appreciate it more, that sterling lecture I gave the other night. Thick fibers, known as the thick myofilaments, and the thin fibers, the thin myofilaments, overlapping with one another. Now, we don't have a, well, I guess if we look closely here, you can see one whole sarcomere. Remember, the sarcomere was the individual unit, and there were many of them that ran the length of the myofibril, I'm sorry. And basically from this pink line to this pink line, that's one sarcomere. How do I know that? Well, because I can see under the white here, I can see all the thick myofilaments, and I know they're attached to the M line. And I can see the thin ones here, which come off of the Z that way, and come off the Z that way, and overlap. And here's your zone of overlap is somewhere over here, where the thick and thin overlap with one another. And as I was saying, a triad, which is a T-tubular with two pieces of sarcoplasmic reticulum next to it, the triads are found at the zones of overlap within a sarcomere. And this is one sarcomere from Z to Z. There's a zone of overlap here with a triad, and a zone of overlap here with a triad. The white stuff, sarcoplasmic reticulum. That's modified endoplasmic reticulum that stores calcium. The calcium that will be released, that will eventually bind to the troponin, that will cause the change in the shape of the tropomyosin, which will expose the active sites on the actin molecule so that the myosin head can grab it and pull it. power stroke. The orange is the T-tubules. And they are an interlocking network that make their way all the way out to the membrane. That's the entrance into a T-tubule that will take the action potential, the signal that came from this, given to this, down the T-tubule, so it can get to every single triad associated with every single myofiber, so that these will all contract at the same time. This is the base. These are some screws.